Everyone, Mr. Nickel here. Just wanted to talk to you a bit about genetics, why I love it, why I think it's so awesome. So I'm going to show you here, this screen here. So there is a bunch of information that is available to us. One of the most popular websites or resources that a lot of scientists will go to find information is this one here. It's called NCBI. What I have pulled up here is the Human Genome resources at NCBI. So there was something a while back that was called the Human Genome Project. And basically the idea was we wanted to create a genetic code, or we wanted to sequence the entire human genome to be able to find, interesting proteins, interesting information. There was a whole political issue that went about it. One of the problems was, who do we actually genetically code? There were a couple competing companies that were trying to do it. One was the US government and one was a private independent sector. What's interesting is the private independent sector, the owner, or sort of like the manager of that, the guy who inspired the project, was said, we are going to genetically sequence my DNA because my DNA is obviously the most important. Classic old white American mentality. But what we've ended up having was this amazing competition to see who could reference the human genome the quickest. And over time, we've actually sped up that process quite a bit. The new technology CRISPR, which we'll talk about in Bio 30 here, CRISPR is one of the ways that, in part, that the coronavirus vaccine has been created, discovered, manipulated, different things like that. CRISPR is an incredibly fast technology. PCR, polymerase chain reaction, was the technology that was used further back. Much slower, but still very effective. And so what we can see here from that Human Genome Project is we can see all of the different chromosomes, and we have sort of a general average of what each chromosome is made up of, what base pairs it's made up of, what sort of amino acids, nucleotides are coding for the proteins present in the human body. And there was a whole bunch of ethical issues that came out of that. One of the ethical issues was, well, if we are going to sequence every single human and have a genetic code of them, can we use that information to make our best judgment about other humans? So for example, we might see that in Usain Bolt, his thigh and calf muscles were designed to be massive and have this explosive raw power. Can we tap into that genetically and make him even faster? And so what it turned into is sort of, if you think about the sports side of things, it would be like, can we use genetics to basically create genetic steroids? So we're not giving them steroids, we're just increasing the genetic composition of them for them to be better athletes. Can we do that? Another conversation that came up was, let's say you are a owner or a manager and you are looking to hire a candidate. Well, is part of that hiring process them giving you their genetic sequence and saying, these are the couple liabilities that I have? So for example, you might see somebody that's prone to cancer at 50 years of age. Well, if you're working at a law firm where usually you stay until about 70 years old, are you going to hire the guy that's going to potentially have cancer at 50, or the guy that will be healthy until 70? And so that whole ethical dilemma of, do we allow businesses, owners, do we allow the DNA sequences to be marketable? And eventually the decision was made. I believe it was President Clinton that said, no, we are not going to allow that. We are going to restrict that and not allow DNA sequences to be marketable. And so what we got out of that was we got all of these chromosomes and pieces of information. So what you can see here, I have another site pulled up. Again, this is on NCBI, but this is a little bit more specific to the GRCH38, which is the code for the human. And what we can see is that there are a bunch of different links here. And what these links will do is these links will take us to the actual sequence of those chromosomes. So we can see the genetic sequence of all of these different chromosomes, all 22 chromosomes, and then also the X and Y chromosome. Obviously, this was a male that was sequenced. And so if I come over here, these are all of the authors that worked on it, different things like that. We can see the organism is Homo sapiens, as we'd assume. I'm going to click this FASTA button here. And what FASTA is supposed to do is it's supposed to give us the sequence. It's supposed to give us the sequence here of that chromosome. It's going to take a little while for me to load here. And what it'll do is it'll give us the nucleotide sequence. We can see that we have only A, C's, T's, G's, and A's. And so what we can start to look for is some of those patterns that we've been talking about so far in Bio 30. Now, you may see this first section and wonder, what on earth is a bunch of N's? Well, N's is considered a non-coding region. And so I'm going to throw this up on the screen really quickly just to show you. A non-coding region is the part before the actual DNA. So this would be the non-coding. Before the DNA sequence, where we have that TA high frequency region, the non-coding region basically serves there as like a protective state. A lot of times, if we remember what our chromosomes look like this. A lot of times we'll find those non-coding regions up near the tips of the actual chromosomes themselves. And if you remember what the term was, those were called the telomeres. Now, what you should also find throughout these chromosomes is you see we had a bunch of ends up there. Let's see how far I can scroll down before I find another region of a bunch of ends. One of the things I also want you to notice here is if you look down in this corner, we have a 36 megabyte file and it's still downloading. This is just a bunch of letters. This is a text file, usually text files. So if you open up the Notes app on your computer and start typing, it'll be like a kilobyte or 3 kilobytes. This is a bunch of letters and it's 43 megabytes. Also notice how long I scrolled and how little down I've actually gone on this page. We can see here another non-coding region, and this is in the middle of this chromosome, which seems a little bit strange. But what you need to remember is that this DNA is swirling around. So when we have a chromosome like this, we might have one strand of DNA that's going like this. And it might go all over the place. It sort of tangles itself up in this chromosome type shape. But what you can see is there's a couple parts of this right here, right here, right here, and right here that those are near the telomeres. Those parts of the DNA need to be protected. And so what we could predict then about, let me scroll all the way back down, this region here is this is probably at either the same or a different end of the chromosome. And this is the non-coding region. Now, typically, or I guess what we thought initially was that this non-coding region doesn't do anything. There's nothing very exciting about it. There's nothing intrinsically different about it. It's just a region that doesn't code for anything. But what we're discovering more and more is that there are things called SNPs. So they're SNPs, and they are small nucleotidic proteins. And what we're finding is that those proteins, those SNPs, are actually coming and binding in the non-coding region. There's some random proteins floating around, and they're coming and binding in that non-coding region. And that's strange to us. But what we think is happening there is that those SNPs, it looks like I wrote the word simp there. Those SNPs are coming and binding and indicating to the DNA that something needs to happen. And so what we're finding is that these non-coding regions are holding way more value than we could ever understand. And so right now we're in the process of figuring out, okay, what are those non-coding regions actually telling us and actually doing? And we don't really know right now, which is awesome. I love that. And then what we can see as well, we've talked about the TATA box, or the AT, high concentration of A's and T's. And what you can see here, there's some C's throughout, but for the most part, this region here is mostly A's and T's. And there are some C's here as well. And so what that would signal to us is that is the TATA box in bacteria, or the high frequency of A's and T's region in us. That'll tell us that that's where DNA replication is to start. And then you can see the rest of the sequence carries on throughout here. And I think that's just absolutely fascinating and super cool. Again, I just want to highlight how massive this file is. If I keep scrolling, so I'm scrolling pretty aggressively right now. I'll show you my, I clicked to my middle button. I'll show you my mouse wheel here. So I'm scrolling really aggressively and we're flying through. And I want you to just look at the right at that scroll bar and see how absolutely pitiful my efforts are. Right now, I've hardly scrolled down through this chromosome at all, and look how much information we have. Now, it is worth noting, I'm on chromosome 1 right now with this one. This is the largest chromosome. It does hold the most information. If we were to go to, let's say, like a chromosome 22, that would be significantly smaller. This page is going to have to reload. Wonderful. Again, we're at a large file, considering this is just letters. But this is part of the reason that I think DNA is so fascinating and so cool, is that we're still learning stuff. We still don't really know everything about this DNA, how it operates, how it's controlled, what sort of mechanisms are working to say, hey, DNA replication needs to happen or it doesn't. We have a wildly creative genetic sequence and genetic code that makes up everything that we do. So this is your introduction to genetics. This is hopefully getting you excited about the content you're about to learn throughout this unit. I love genetics. I think they're fascinating. Hope you enjoyed this video, and we will talk to you in the next one. Peace.